I'm Dr. Anupma Harshal. I'll just give you a little more information about myself. As an independent woman in STEM, I work for several prestigious Government of India projects. I have done a lot of undergraduate research while I was in the university. I've been a teacher for 17 years at Bombay University. And then I decided that this is what I want to do. So I've been in the domain of science communication and public engagement. I have a wonderful team of collaborators. That's Dr. Anupma Sharma. He's a consultant and the director of Lister Cancer Hospital. And he also handles, he's a major in testicular cancer. That's Dr. Ashwini Nandoskar. She's a renowned neurologist at the Imperial Health Center, again, NHS. And I have a wonderful teacher here, Dr. M. Bharata Jyoti. She is an assistant professor at a beautiful college in a city called Vijayawada, which is where this study was conducted. I don't want to thank people who made me stand here tall at the end of it, so I'd like to do the honors in the beginning. I'd like to thank Prakash Lab at Stanford University for all the foldscopes that I have been using since 2017. I will just show you that in a minute. I'd like to thank STEM peers for having given me this grant, which has allowed me to travel pan-India and do these workshops with young adults, because that is where I see a lot of potential. I see the change makers being there, and they are the ones who are really going to be caregivers. They are going to be researchers, innovators, and therefore I really work with them. What's the first thing that comes to your mind when I say the word foldscope, and what would foldscope have anything to do, with an antimicrobial resistance? This, my dear friends, is the foldscope, and oops, I just dropped it. It's a paper folding microscope discovered by a physicist called Dr. Manu Prakash at Stanford University. If you didn't know about this, it's good to hear about this, okay? This is made out of paper, so you could dip it in water 13 times, and the 13th time it will give away. So this foldscope particularly I have been using since 2016, and I hope you can see it has gone through multiple hands, but it still remains absolutely going to be talking about what is she using this for, what exactly is she going to derive about, and connect it to a conference like this, especially in the area of science communication and public engagement. I would say a domain which is not tapped to its full potential. Thanks to COVID, we all had to actually ensure that we not just work in our labs, but all that we did in our labs actually reached the common man for them to engage in those practices, imbibe those practices, and make their lives and the world a better place to be. So essentially, this is more about my table of contents that I'll be talking, but I want you to focus here. Can anybody guess what this is? Anyone? I'm not so sure if you'd like to hear this, but this is typically an onion cell. An onion peel or sheath that has just been taken off, put on a slide, and has been mounted by a 6th grader who does not speak English. So science, messages, impact on public health doesn't need a language. It needs a mind, and it needs a message that can go from this heart to that heart. And I hope at the end of my session, I have made a difference to your life. This, ladies and gentlemen, is the nucleus, if you can see clearly here. And just see, no staining, absolutely none. And this is 140x. But thanks to a camera that you have, so in all my lectures, I don't tell children to put away their cameras. I tell them, please keep your cameras, because you could just keep your camera on this foldscope, and I'll just show that to you in a minute. So all you need to do is you need to just keep your camera on, and you need to put your slide here. And if I can actually do that for you, I would be happy to show that to you. So I just need to, 1 second, my memory is full, but I'll still give it a try. So all I need to do is whatever is there inside, I can actually put my camera on it. Sorry, I just have too many things in my hand, but I'll still try my best. So I can just keep my camera on it, and give me a minute, you'll be actually able to see what is inside. Can you see that?
Yes.
So when I go to villages, when I'm talking to tribal children, to indigenous populations, I don't need to tell them there is a parasite there. I don't need to tell them there is a fungi spore growing there. I can actually do it show it, and that's where the child gains confidence that he learns best when he does it with his hand. So this is what my study is all about. While you may find it a little odd that I'm here, but I have some valuable data to share, and that's why I come this far. So this is version 1 of the foldscope, and there is also a version 2 of the foldscope which goes up to 340x. And by the way, there's also a version 3 which goes up to 3000x. And believe me, none of it breaks because it's made out of paper. So this is frugal science. And these are my findings that I will be presenting to you today about a workshop that I conducted in Telangana. And what exactly I do in these workshops, what is the change that I'm trying to build. I follow Mahatma Gandhi to the world. You can't expect everybody to change. So be the change that you want to see. So I chose to quit a permanent job in a university, and I went out and I told the government of India that I'd like to do things which would help them improve the lives of lots of young people who are there in the country. So I have a question for you. I give you 2 minutes. Can you think of a superhero that you like? But wait, you have to tell me the reason why you like him. Just think of a superhero that you like and give me a reason why you like him.
Mandela.
Why?
He brought peace in South Africa, reconciliation after all struggles. He went on without complications.
Thank you. Thank you. Just remember the reason because we're going to draw something. And there's a disclaimer here. I'm not here to teach you or preach you because I don't think I look like either. So this is going to be more of an exchange of ideas. Anyone else? Anyone else? My superhero is my dad, my mom. They're amazing problem solvers. I can just look up to them. I can just ask them, I want to go to Paris. How should I go? So my dad said, hey, just go to London, take a Eurostar and then go to Paris. Yesterday I met another friend, Vinita. I asked her, hey, how do I go to Eiffel Tower? And she said, hey, Anu, take the train from here, go walk it up. I met Anil there and we again chatted. So essentially, these are my heroes because they are helping me. They're problem solvers and they bring to the table an amazing set of qualities which inspires me. And wait, it helps me as a population. That the moment he told me how to go to Eiffel Tower, believe me, it didn't stop there. I told another 2 delegates, do you know how to go to Eiffel Tower? They said no. And I said, hey, just walk down here, take the metro, where he came, just get down there and you'll find the directions. Do you think that's how bacteria communicate to each other? Do you think that's how bacteria communicate to each other? These are just a few superheroes, but I think I love my mom the most. She's no more. And I stand tall because at first I wanted to put a picture of just a mom hugging a child. Essentially, these superheroes have qualities which are actually helping the population. And it's so simple for somebody like me to tell another colleague or a delegate in a conference that, hey, this is what you need to do. Let's look at bacteria. Aren't they really doing the same thing? Aren't they really doing the same thing? So they have a beta-lactam which challenges them, their existence. They come out with a mutation, select the mutation that helps them break the beta-lactam, and then they say, hey buddy, why don't you just take this know-how? You can call it a plasmid, you may be calling it an R plasmid or a col plasmid, but that's essentially, let's not go into the molecular biology of it because that's not what I'm here for. But essentially, that's how resistance is moving across the population. And by the way, those who have gathered the resistance are actually their superheroes too. Okay, so let's not be harsh on them, but we choose to name them as superbugs. Okay, so that's a little introduction of the domain area that I'm going to be working at. Essentially, each superhero, whether fictional or real, represents different values and qualities that inspire and influence people in unique ways. So while you said Nelson Mandela, even I have a lot of respect for him because what he's done, nobody else could even dream of it. Innovative problem solving because they use creative and innovative solutions to overcome challenges. They give you solutions to problems that you would never even think of. They will give you ways and means that will just not strike you as an individual. And that's essentially what a superhero does. And that's essentially what a superbug is also doing. So when I teach my students, I don't tell them bacteria are bad. I ask them, what would you like to see in a picture, which has animals, plants, pigeons? And they say, ma'am, I think coexistence. Coexistence. Bacteria didn't make antibiotics for us. Bacteria made antibiotics to inhibit growth of other organisms which were infringing on their territory. If I'm wrong, you are free to correct me. So essentially, we exploited this domain to our advantage. That's how we've used it. Of course, it was in the intention of the well-being of mankind, and so we have continued to do so. Well, during my sessions, I ensure that I run a hashtag called Know Your Foe. It's important. Yes, it is. Because you couldn't have taken an antiviral drug for a bacterial infection. You can't take an antibacterial drug for a viral infection. You can't take an antiparasitic drug for an antifungal, and so on. But it's important for this young, young people, I would say, in the age group of grade 5 to undergraduates and postgraduates, to understand that who's your enemy. If you have a fever, like I was just talking to somebody, they just said in my country, you can just go to the medical shop or pharmacist and tell him, hey, I have a throat infection. And by the way, he's not a medico, but he'll give you the right medicine and the dosage for 3 days or 5 days. And I'm sure this is happening, in a lot of countries. Probably it doesn't happen in A-lister cities like Delhi, Bombay, Chennai, and Calcutta, but it happens just about everywhere. Okay, where the pharmacist is more the medico than anybody else. Essentially, what is happening is another way of handling this. So my mother, when she would have fever, on day 2, she would feel better. Although the doctor has said that, let's complete this dose for 3 days, 5 days. And I would say, no, please don't do that. Complete your dose. And she would say, no, I'm fine. I'm fine. And this is what I ask children, is this happening in your house? And they come back and tell me, yes, it's happening in my house. And that's how the drug gets left over. And that's how you have the same drug next time for an infection. Now, whether it's going to act or not is something we're all here to discuss, and we know what AMR stands for. So essentially, I help them to identify or at least resonate that fever or any kind of illness could at least have these 4 initiators, okay? So bacteria, viral, fungi, and parasite, and therefore you have an antibacterial, antiviral, antifungal, and antiparasitic. So know your foe is very important before you actually start a treatment line or a protocol. Yes, essentially what do we do in our workshops? We introduce the foldscope to an AMR workshop attendees so that the hashtag know your foe begins. These are children who, like I said, there's a lot of indigenous population that I work with. They don't understand English. But you know what, when you tell them make a slide, give them a glass slide, or I could even show you, if you give me a piece of paper, how to make a paper slide. You can just fold a small piece of paper 2 times, make a hole in between, put a sellotape, you have a surface. And I tell them, please don't take sample like you take your chutneys and sauces. Just take the sample, the size of a pencil tip, and then put another sellotape, mount it there, and see for yourself. See for yourself. Ask questions. Ask for evidence whenever you're looking at things in life. And that's exactly what the young population. I don't want followers. And we're not those kind of leaders who want followers. I want innovators. I want children to ask questions. I want children to ask odd questions, handle different types of life and then kind of visualize. So they actually examine samples of onion, potato, colon, and squamous. And this list is endless. And why did I begin with the onion slide? Why did I begin with the onion as a sample? I'm sure somewhere we've all been students. If it's easy, if it ain't simple, I don't want to do. Right? So taking out an onion peel is very easy. You just take out a peel, keep it there, put a sellotape, and make your observations. And that was exactly what I showed you when I showed you that slide. So these are onion cells. This is the nucleus. And you can jolly well see that without any staining, a 6th grader has taken about 8 to 12 minutes. And you know what they do? They struggle to make these slides, but they come back and show you, hey, I've observed this. And what if there's no sunlight? What if there's no sunlight? This light is enough for you to visualize in the foldscope. What if you didn't have this light? Just buy a $1 or a 50-cent torch, put it in an Amazon box, an old Amazon box, and ensure that the work goes on. You don't have glass slides, let's not complain. I'll have to make a paper slide, but let's encourage this Gen Next to actually get going with what they have to find out. First, identify their own problems. And second, not wait for anybody from Stanford or Oxford or for that matter, any other university to come and solve their problems, but to generate their own solutions and come back and solve their problems in their local region. With no offense meant to these institutes, I have a high regard and respect to them. Make your observations, draw, look for characteristics in cells and take images. Like I said, 7th to 10th graders are not allowed a phone in Indian schools, but undergraduates are allowed phones. So they have to take pictures. And does it end there? No. There is a community site called as the Microcosmos site. where you can post in Tamil, you can post in Brazilian, you can post in Spanish. If you're a little shy of writing, just post your pictures. Hey, I saw this rod. I saw something moving in my water sample, and somebody from Berkeley will say, hey, that's a rod for. Do you think those are communities for? So my children are not interested in Instagram, but they like to continue on communities. So every day my inbox is filled, and I show you what these students are doing, because at the end of the day, they're liking what they're doing, and therefore they're really enjoying it. So they draw, and it's a completely open-ended inquiry. I don't tell them, hey, do this, do that, take an onion peel, take a potato, smash it, remove all the pulp, just observe and keep going. Then you tell them that if you know your four, what is going to be the to-do and the not-to-do list when you are, handling an antibiotic or an antimicrobial agent. And that is where they come up with solutions. And that is where, my dear friends, I think our heart lies. Not in telling people the solutions, but helping them to generate their own solutions. And they'll say, ma'am, next time I have fever, I'm going to indulge in TLC. And that's tender loving care. I'm going to have warm food. I'm going to have lots of hot liquids. And I'll wait for a day or two before I go to the physician. Somebody else came and told me, ma'am, would it be a great idea if my mom is flushing down the antibiotic? Is it not going to put some stress on the soil? And I said, why don't you look up a book on sewage treatment? Do we have sewage treatment protocols which allows us or tells us what to do with leftover antibiotics? At least our textbooks don't have. So I'm sorry, if you have a better version, do just share it with me. But this is where I get them to think and tell me what's the solution. Okay, because these are going to be the people who are going to be caregivers. They're going to be the people who are going to be partners. They're going to be people who are going to be in the public health domains, and they're going to be the people who are going to be making policies to understand what they need to do. Yes, so I am really happy, and I hope that you're going to love and kind of enjoy what you see. So this is no harm was carried to the plant. So we took a sellotape, we just tapped it on the flower like this 3-4 times, and we mounted it there. Can you identify what this is? Well, this is a pollen. Yes, this is a pollen grain. You must be wondering, because most of you are very learned from a medical fraternity, what would these cells help individuals and how would they help them to really understand the concept like AMR? But when I see cells, when I see different cells, I can understand what a salmonella looks from a clavaria. I can understand what a staph aureus looks from a bacillus cereus. And this actually, because I'm doing it, I'm able to understand differences. I'm able to understand membranes. I'm able to understand outer covering, the nature of which and how cells interact with each other. So that is just taking a sellotape, tapped on the flower, so no harm was caused. That is also to tell you that this is how these sessions are carried out. I take them in a garden. I ask them to kind of look at what is around, take one leaf. So today I had a very amazing gentleman in the canteen. He'll be here when we are being served lunch. I just asked him for a leaf sample and he gave me a little bit of salad. I took out a small film and you can actually see the stomata on it, So it's a eureka moment when they say, wow, that's like a pollen. Oh, is that a pollen? And then they draw it and then they write their own explanations for it. Sometimes I do set up cases. Okay, so I set up a case where I have a murder scene and then I tell them, hey, come on now, let me tell you what's the investigation. And they say, we'll ask for a hair sample, we'll ask for some sputum sample, and we'll ask for some fiber sample. Now, obviously, I'm not in a clinic, so what do? So we take the sellotape, tap it onto different surfaces, allow them to visualize the fibers. I allow them to wash their hands because I may have access to a hand washing place. I ask them to take their squamous cells. Well, I also ask them to take hair samples and they pick out the facial hair and the eyebrow hair. And the 4th grader came back and told me, he didn't know plastics. He came back and told me, this hair is very soft and it has less color. This hair looks like a sword. It is very sharp and it has lots of color. And that's what helped their teacher teach them about plastids and therefore photosynthesis. So not going much into it. Essentially, we provide them with these samples. They make their own slides. And believe me, sluggishly motile is what we must say because they take about 3 minutes, 5 minutes, 12 minutes, 20 minutes to get going with the slide. They are not trained histopathologists or path labs, but once they've got it, they know that the charm is not in visualizing or seeing in the microscope. The charm is in actually preparing the sample, and that's where they get the hang of it, and that gets them enthused to take this further. We then encourage a discussion and questions about cells, their different characteristics. That's the same picture for you. So this is how it looked. use the phone, and that's almost a 340. This also tells them about arrangement of cells. I hope you can see this looks, for most of my children in India, this is a brick wall. Okay, where the bricks have been laid very nicely. And then they start imagining that, oh, are all cells like this? And the answer is that I don't know if all the cells are like this. Let's see what a potato sample would look like. And voila. They start from here. I hope you can see this. Really crowded. Yes, and as a learned teacher or as a learned person or as a guide or a facilitator, you tell them, hey, there are too many cells there. You can't see the cells really. And then they come on to something like this. And this is the clarity of cells, which again gets them to the space of saying that all cells are not arranged as a brick wall, but cells have different arrangements, which again helps them to visualize cells, whether it's in a pathogenic infection or whether about their own multicellularity. Yes, those are tomato cells. And they'll come back with questions saying, why do tomato cells and potato cells not have nucleus? And then they come back with the answer saying that, oh, they are actually storage cells. So during their course of storage, somewhere the nucleus actually gets enucleated. Okay, so getting them to seek answers. This, of course, I'm sure you all can identify. Yes, the stomata, again made by a 9th grader. This was at Ashoka University just about 20 weeks back. And this is a dirty water which we took from the pond very close by. We had to do some jugglery and get this water. I really don't know what this is. I posted up on the site waiting for somebody to help me identify what it is. But there was a green ring on the bottle of water that we had taken out. And so it's on the side waiting for somebody to explore and let me know. Yes, these are the collages that fill my inbox every day. And I hope you can see I encourage them to write. A lot is being said about the Gen Z, but unless you put it in pen and paper, unless you put your thoughts there on paper, it's not going to happen. So they say, my drawing is not good. Shall I type it out in Google Doc? And I said, no, can you make a little effort and draw it? And that's when they get to the space. And this is, of course, not one of the best ones, but I have beautiful hand-drawn diagrams where the child is able to actually put it on paper. This, you can see, is a fingerprint where I give them an investigation case to find out who the murderer was. These are fibers. This, of course, is again an onions cell. These are pollen. This, of course, you can see this is a hair tissue. Again, this is a fly wing, and these are fungal spores. Again, this is a fern rhizome which has been stained. So these are the type of collages they make, and they send it to me every day, saying that, ma'am, we today explored whatever vegetable mom has made. I've explored this, and this is what I'm going to be posting on Microcosmos. Essentially, my role here is zero. I've just got them initiated to ensure that whenever they are doing any piece of research, any piece of answering a question, they report it somewhere and it is tabulated. So essentially, this is not my way. This is essentially what the children are taking forward on their own go. Yes, so this was 400 girl students, and they were from grade 11 to BSc. And if you can see, the subject combination should give you an idea. This was commerce, economics, and civics. So these were non-science students. There were just about few students from BSc, which was about 60, but out of the 400, all of them were from grade 11, BA, BCom, but a lot of them were from commerce subjects. Again, trying to tell you that science is not only for science students, science is for everybody. We have to get everybody, as a nation, I think all of us will agree. Yesterday, there was a speaker who said there are 40 nations here. As nations, I think we have to get into the space of developing a scientific temper. And that is why I think the United States and the United Kingdom are where they are in terms of research. And a lot of others who are not there are looking up to them for the kind of research and the research culture that they propagate across the universe. Yeah, so essentially, these were students who were, very committed. They almost kind of spent 12 to 20 minutes to make the slides. And, the engagement was driven by curiosity and the novelty of the foldscope. Yes, after we did the foldscope workshop, just about within an hour, we shared a questionnaire with them. And during the course of the foldscope workshop, I run the same questions. Who's your superhero? And why is your superhero your superhero? What's the reason for it? And also, do you really need an antibacterial agent in the hand wash that you have? Because many of them answer that, man, soap anyway kills the bacteria, right? Do I really need an antibacterial agent? So I leave it to them to understand, make valid choices tomorrow when they go and pick up the products, so that at least somewhere they don't contribute to this market coding or mentality of just buying what is being advertised, but use their rationale that soap is anyway doing its job. Do I really need an antibacterial agent in the soap to, rid. And I also get them to, ask questions or think in the zone of, are bacteria really bad or naughty? And they come back and tell me, no, they're not naughty. They're there all over my skin. They're there in the curd that I eat. They're there in my gut. And somewhere they get naughty when our immunity is less or they're pathogenic. And that is when we have fever and different diseases. So not showing any of these bacteria in a bad light, but saying that if coexistence is the model, everyone has a right to kind of, this is typically, introducing them once they have identified, once they've seen the different cells, and then I take them to the space of, identifying that these are bacteria, viruses, and you'll be glad to know that you can see up to 1 micrometer in the foldscope, and I think that's amazing. And there are children who won't understand what a 1 micrometer is. So I tell them, do you have a scale? Do you know what 1 millimeter is? And they would say, yes. Okay, if I take this distance as 1 millimeter, can I divide this into 1,000 parts? They'd say yes. And then I ask them to imagine, can you now imagine one part out of this? Which means 1 upon 1,000 of a millimeter is a micrometer. If it ain't simple, I don't want to learn, okay? I am not a special education teacher, but I have a child who's a special educator. And I think that is what made me be empathetic and, accommodate diversity in my classroom. Well, there are a lot of, outcomes that we have, from a workshop like this. And there's a lot of data here that I can share, but that's not what I'm here for. I'm just trying to tell you that questions like, are microbes stronger than your, other cells? What happens when what is the confidence of using a microscope once you've used a portable microscope? How do you read scientific literature? So if I have a organism or a cell, what do? I go and look for literature. I'm going to find out what the cell is all about and then kind of, really take it further. Picturization of the cell, knowledge of getting, a lot of content through this. Essentially, then discussing with them how AMR is actually developed in these organisms. So then it becomes very easy for them to understand that, oh, because bacteria are different, therefore you're using different agents to, combat different types of fevers and diseases. And essentially getting them to understand how easy it was for one, superhero to transmit through lateral transfer. That's how AMR is transferred. Also talking about, mechanisms in developing new strategies to counteract resistance. And these workshops provide visual insights into how bacteria would respond to antibiotics, reinforcing their theoretical learning. This is what my classroom typically looks like. And this is in the Telangana college. You can see again, not a great setup. This kind of workshop can be just done anywhere in a classroom setup. I really don't need much requirements for it. Typically also, making them understand what this health crisis is all about. It's the young adults who need to understand that the global health implications of AMR to advocate for prudent antimicrobial usage. Raising awareness through workshops can drive community actions and policy changes to combat AMR is the space that I come from. And that is why I thought it would be appropriate for me to share my data here. So I'm thankful to Magnus for having accepted my data set. Yes, many of them will come back. What is our country doing? What is what is WHO doing? What is CDC doing? And yes, there are a lot of efforts that are going on. So we then, they're more in a receptive mode to understand what are the different policies that have been made for them. This kind of public education and engagement are essential components of a global AMR strategy. Young adults contribute by participating in awareness programs and promoting responsible behaviors. And that's precisely like, what I began with, that I'm not here to teach you or preach you. And therefore, yeah, so this should be a few of my last slides. This is how the newspaper coverage in local newspapers in the local language helps me share this message across a large population. And that's how I get to, conduct more workshops and therefore populate the message further. This is the photographs that you can see young children from grade 11 with my collaborator, Dr. Bharta Jyoti. And empowering the youth with knowledge and tools fosters a proactive approach to combating AMR. This, of course, is how the world is, moving towards reducing the antimicrobial resistance. And remember, if you can prevent the fire, you'll never really have to use the extinguisher. So thanks a lot for your kind attention. That's me. And typically running on power of coffee and mitochondria. I'm neither a leftist nor a rightist. I'm a STEMist. And selfie is what, most of us love to have during our sessions and the work we do. That's me performing a Kathak dance, saying that you're confident yourself, and that's how you pose yourself to the world. And this, of course, is something that I love because I think now nothing in life is to be feared, it is only to be understood. And now is the time to understand more so that we may fear less. Thank you very much, ladies and gentlemen.